All right, so we're going to move to our next agenda item. It's a presentation of blue-green mitigation strategies and technologies. And this will be given by Dail Laughinghouse, who is with the University of Florida.
But anyways, so yeah, and I talk fast, so I'll try to get this at least through this. And then, yep,
Okay.
who will be changing the slides for me? Will somebody be doing this?
Yes, you just tell us next slide and we'll advance.
Okay, I will do that. Well, anyways, thank you so much, Jennifer, for inviting and allowing me to do it via Zoom. I wasn't able to put back-to-back meetings on the Gulf Coast, but there's a contact for me here for anyone that wants to meet with me in person and talk longer, because I do like to talk and I like to listen to. Anyway, so we're going to talk about cyanohab mitigation management. And as Jennifer said, I'm a professor at the University of Florida, based at our Fort Lauderdale Research and Education Center. And I spent the last 20 some years working with cyanobacteria and the last 10 years doing a lot of mitigation and management. And in Hab science, the part of mitigation and management is probably, it is the part, well, I know that Barbara's in there. So, and health aspects are the parts that are most pressing and I guess least knowledge known. So please go to the next presentation. So just to have a general outline, we're going to talk about blooms, proliferations. I will not talk about crusts and mats, but crusts and mats also can have toxins. We'll talk about some proactive management methods that are used and some of the data behind why they are used, and then some reactive management methods that you may have seen. But yeah, like I said, in 20 minutes, I will do my best. Next slide. So what is a bloom? So I will focus on the treatment mostly of planktonic blooms, and I am talking about cyanobacterial blooms, which are the blue-green algae. I always like that little thing on the top right corner. When I give presentations around the world, actually, it's so you want to visit Florida with this beautiful, green scum, which I actually love. I think it's beautiful. But anyways, what is a bloom? In this case, it is a dense growth of algae. We hear about them because they can produce toxins, and these toxins. What are these toxins? These are neurotoxins, hepatotoxins, and then sometimes dermatotoxins. But if you look at Ingrid Kors in the last WHO update, she removed dermatotoxins from toxins because they don't really kill people; they just cause a rash. So let me not say kill people. Let me say they don't. Mortality in animals, including humans, is not really caused by dermatotoxins. The other part that many people do not think about is that they're able to harbor pathogenic bacteria. So, okay, even though they may not be toxic per se in a microcystin, Pseudomonas antitoxin, saxitoxin, they may have Vibrio, they may have other types of bacteria that are pathogenic on their mucilage. Okay, and they also contain taste and odor compounds. So if you've ever, that smell of earth that you smell when it rains, that done by the actinomycetes, that's also produced by cyanobacteria. So that's geosmin is the earthy smell, and if you smell an insecticide smell, that's called metasborneol. And those two compounds can get into drinking water and really cause havoc because no, you don't like that taste. It can also be transferred via milk if the cows are drinking from areas with blooms. And also there are some notations that it actually goes from the chicken to the egg. So so there's a lot of things with geosmin. That's another part that is that some area of research. And also treatment is very difficult of these because they're very small molecules. And these have been observed for centuries. Blooms have been on for the coast of Florida on the red tide for hundreds of years, and blue-green for their data that go back and say some of the mass extinctions of dinosaurs were due to huge blue-green algal blooms. But let's not get into that. I'll take forever with those conversations. But please, next slide. But and these are just some beautiful things when you see the blue, when you see that those types of like scum, you can't, someone will ask me, what is the species? And I said, I have no idea, because you need to look at them in the microscope. So these are just showing the beautifulness of what they look like. These are, there's, just on the top left, that's the Dolichospermum species. On the bottom left, you have three different microcystis species. And then on the top right, you have a planktothrix species. And then you see on the bottom right, there is a Sphaerocomopsis, that little curly one that looks like a pearl necklace. These are actually from the West Coast, so close to where you are. That one produces guanotoxin, which is a very, very potent neurotoxin, but degrades very fast, so we can really never see it in water samples. And also, there are no standards for it. But hopefully soon we have it growing. So next slide, please. But planktonic blooms aren't the only issue. And on your side in Florida, and then also on the marine side on the West Coast, you're seeing a lot of benthic blooms. And these are an issue worldwide. They do produce compounds and toxic compounds. Next slide. Including the ones found on your coast, but I will not focus much on this. But when you see these kind of clumps floating, Charlotte Harbor, Tampa Bay, there's a lot in Matlacha. So yeah, these are also producing toxic compounds. So continue. But now we'll go with what we're actually talking about today. So we're talking about management. So I always like to have this, there's a bloom and now what? A lot of research has been done on ecology of, why these blooms form, but you ask a homeowner or a government official, they're like, okay, I got it, they form, but what do I do now? Right. So again, some beautiful blooms of cyanobacteria in here, and these both are actually in Lake O or the Lake O Rim Canal. Continue, please. So I like to separate blooms in this way. So you have proactive and reactive approaches to bloom prevention and treatment. And then they can be kind of separated into chemical, physical, mechanical, and biological methods. All this red you see are what I will be talking about in the next couple slides. I think that this I published this, and I can send this to you guys if you want to. But we're going to focus a little bit on nutrient loading of why people say decrease nutrients, some about P binding compounds and why, colorants, which are dyes, that's on the proactive side. And then on the chemical side, we'll focus on algicides and algicides. And then on the physical part, we'll talk a little bit about artificial mixing, and then ozonation on the reactive side. I removed all the biological part because there's a lot of snake oil there, but there's a lot of potential in the biological area. And there are a lot of projects being done, inclusive there are projects with red tide that are looking at biological techniques. Continue. So, why do people say reduce nutrients? I mean, most of you guys are scientists or science literate in there. So I heard about all these wonderful projects and saw one with SAVs that really clean water. So, if you understand, so the reason is that and in general, the idea is that phosphorus is limiting nutrient in fresh water. But Florida, we have a lot, right? So cyanobacteria are P limited. The idea is to decrease PN and input, but there's also legacy phosphorus and legacy nitrogen if you talk about what Carl Havens did. So if you look at this paper by Hans Perl that he presents everywhere he goes, it is called It Takes Two to Tango, and that's the bottom right. You see the N and P dancing. What you see with this manipulation above control, that when you added N and P and these whole-late nutrient treatments, you saw a 500% increase in blooms. So, and the same thing with the slide up on the top right, with the graph up there, as you see an increase in TP, so that the log on the bottom is total phosphorus. If you look towards the end, when you have an increase and you look at percent total phytoplankton biomass, you see that cyanobacteria really increase when there is a lot of phosphorus. Okay, and why is that? Because many of the cyanobacteria can fix their own atmospheric nitrogen. So again, on the proactive side, we're looking at decreasing nutrients, but this takes time, as you guys know, and a lot of times, even if you decrease the nutrient input in a system, this is freshwater. Remember, talking about freshwater, blooms will happen for 100, 200 years. So this is why a lot of reactive management are still needed. And of course, again, like I said, this is highly dependent on the water body. Different nutrients drive different algal groups. So even though, and I removed it from this slideshow, is that even though you may have a decrease in phosphorus, you may see an increase of another cyanobacterial species. So it's not cut and dry. But again, in general, of course, we do know we need to clean our waters. But another aspect is a lot of waters that have been cleaned around the United States, you're seeing an increase in benthic blooms because they're very tightly correlated to the benthos. But that's besides the point, just an extra caveat there. Next slide, please. On the mixing aspect, so if I don't know, again, if what projects you guys have funded and what you guys see day to day, but you probably do see a lot of aerators or mixers in ponds, especially if you live with HOAs, which all of us probably do. Anyway, so mixing is is there to prevent scumming. That's that bloom formation, and it prevents stratification. It prevents the water from having a top and a bottom. It really mixes that water, and this circulations to select for better algae. What are the better algae? Are the diatoms. Because cyanobacteria, as they can float up and down, when the water gets stratified, they really stay at that top and form that scum. So if you look at the slide, the graph here, you're looking at time of season and increasing with mixing. So when there was no mixing, you saw there was a lot of cyanobacteria as this season went on. So you're looking at March to October, and this was in Dutch. I think I see the OKT. So I have to tell Peter that her graph from her paper in 90s was in Dutch. Anyway, so you see that towards summertime, when you did not mix, you saw an increase in cyanobacteria, which we see, so warmer weather. In this case would be, after the rains and the more rainy season here, you see the increase in cyanobacteria with no mixing. But when you mix it, you start seeing diatoms dominate, which, okay, that's wonderful because diatoms, in this case, for fresh waters are not producing the toxins. For marine waters, you do have a diatom that produces domoic acid. But that's, again, this is just reminding you that freshwater and marine, even though we have HABs in these systems, they are very different and produce different things and are caused by different aspects. And control is even different. So anyways, so including in the mixing part, there is a thought about what about phosphorus resuspension? So if you're mixing the water and you're mixing that benthic layer, all of a sudden, if you turn off the mixers or aerators, you now have that phosphorus in the water column available for cyanobacterial growth. So again, it's a very important design of these and understanding the system because it won't work for all. Again, but this is proactive because you're going to be doing this all year long instead of applying it after bloom forms. Go ahead, next slide. So a lot of, especially in golf courses, you see, and probably in a lot of your HOA ponds, this won't happen in a whole like Lake Erie. You're not going to put blue dye everywhere. People use aquashade. Aquashade is when I say a compound, a product is one I've tested, not saying that's the product one should use. But these are blue dyes, and in this study that I have on the right was work that we did that was published earlier this year using aquashade. So blue dyes are used in loaded systems, and then they control. So these dyes control by competing with the photosystem of, in this case, cyanobacteria, because if you look at where the wavelengths, about 550 to 650 nanometer range, where this blue dye uptakes light, if you look on that wavelength, you see, oh, wow, that's around phycocyanin and phycoerythrin. So phycocyanin and phycoerythrin are cyanobacteria-specific pigments. So that's the theory behind it. So when you go look at the graphs here, this is 1 month after treatment is A, 4 months after treatment is B, and the black is cyanobacteria, and L36 and L17 are two HOA ponds. You see mostly at 2 ppm or over time, you do see a decrease in cyanobacteria. So in this case, this is the initial report of looking at this. And so we do see anecdotally and also here that decrease in cyanobacteria. I'm sorry, something fell in my office. A decrease in cyanobacteria with the use of dyes as a proactive treatment. Okay, continue. Another thing, and in this case, is not an algicide. This is a nutrient control. But remember that if any product or device that is stated or intended to be used to kill an alga is a pesticide and must be registered under FIFRA. Okay, so in this case, it is trying to lock phosphorus to decrease the availability for bloom growth. And here was a slide from a study done by an industry colleague and looking over time, and really saw that after a year and using Phoslock, that they did have a decrease in the bloom. But this, again, systems are different. If you have resuspension, more phosphorus come in. But anyway, so this is not a one-time only application, but it's part of a management plan, integrated management plan for nutrient control. Okay, and there are other products now, and actually, I don't know if, can you go to the next slide? Yeah, I removed that. So what we also noticed was that Phoslock can actually sediment microcystin at high concentrations. So sometimes with algicides here, which I'll start talking about reactive, you can do a shock and lock. So you can shock them and then lock the toxins. Okay, so that's again more of a integrated management system. But going back to products that work. Algicides have been used extensively and are used extensively in Florida and around the world. And then I think since the 70s that copper's been used. Peroxides are more recent, but there are many U.S. EPA registered algicides for cyanobacterial control. What you most have are copper-based products and peroxide-based products. And copper-based products, not all copper is the same. Though elemental copper is the same, but these are all formulations. As you know, think about medicine. When you have a generic compared to a regular and another generic, you see there is a bit difference in efficacy. And so you do see difference in efficacy in these different products. So with copper, I'll go there first. You have copper sulfate, copper ethylamine complex, and copper citrate and gluconate. So the reason is that you have chelated coppers and acidified coppers to be able to work in different water systems, like acidic water systems, hard waters, etc. And then on the peroxide aspects, you have sodium carbonate peroxyhydrate. That is the granular peroxide. And then you have a hydrogen peroxide and peroxytic acid or peracetic acid mix, and that is the liquid peroxide. Okay, again, remember that any product or device that claims or implies the mitigated pest must be registered under FIFR by the US EPA. So next slide. So how do copper-based algicides work? So again, they're used a lot. People are moving away from them because of just accumulation of copper in systems, and a lot of waters do not allow it because of this and the off-target, non-target effects that copper has in systems. But anyways, how does it work? Actually, don't fully understand, but in theory, it disrupts electron transport, it prevents cell division with glutathione. It inhibits enzyme catalase and other enzymes, free radical susceptibility, photosynthesis, P uptake, and nitrogen fixation. So it really does a bad wonder on these algae. It interferes with cell permeability and binding of essential elements. It can penetrate when they add these acidifiers, when they add these adjuvants and other aspects. That's when it's able to penetrate the mucilage, get into the colonies, and really affect these cyanobacteria. And it's especially good with the benthics because peroxides will actually sometimes cause benthics to grow. Decades of use, there are many restrictions now, and many countries don't allow it. Like I do a lot of work in Europe, and it's not allowed anywhere in Europe, except like in Turkey and in Greece, they allow it. So, anyways, but you know, the Netherlands, the Benelux countries, a lot of the Western Europe don't allow it. Neither does the UK. Well, the UK may now because they're outside of the EU water framework. I haven't seen the new updates for the UK. But remember, not all waters are the same. pH, alkalinity, temperatures, hardness, etc. They really affect efficacy. So next slide. So that's copper. So let's talk about peroxides, and then I'll go get into some examples. So peroxides they affect cyanobacteria more than eukaryotic phytoplankton, and that's due to the Miller reaction. So in the regular and so they're during photosynthesis, the eukaryotes produce peroxidases. Aces will degrade peroxides. They produce that during photosynthesis. Cyanobacteria don't. They have a Miller-like reaction where they don't produce peroxidases. So what does that mean? That means that that the peroxides are more susceptible by like 100 times to the cyanobacteria, so they will kill those and allow the other algae to exist. But what also is another caveat of there is if you have a bloom very mixed with eukaryotic plankton, with diatoms like green algae, etc., those are producing peroxidases, which will then degrade the peroxide. So you have to use higher peroxide levels. But also certain bacteria can produce peroxidases. So this is where you hear the term microbiome, phycobiome, cyanobiome, and how that actually is important to understanding why these things work, why these things don't. So that's why there's no one treatment works for everything. So anyways, so again, peroxides, they break down into to water and oxygen within 24 hours usually. So they're relatively safe to non-targets, but application rate does matter. So you have to follow that label on the bottle. Again, so what we've seen and others, within a few hours of treatment, you have 70% decrease in photosynthetic vitality, 99% collapse in cyanobacteria microcystin concentration within a few days. But they are more effective at high light intensities, and they are less effective at low NNP concentrations. Next slide. So here is a example of product evaluation to look at bloom and of Microcystis aeruginosa in Lake Okeechobee. This was, again, if anybody wants some of these papers, I can send them to you. What's in red are peroxides, and the G's and L mean granular and liquid. And then the other ones are the cutrine and algae. The two, you see the two top are coppers. There's another copper, which is C clear. And then there is Captain XTR, which is a copper again, and then an endothol, which is a hydrothol. Okay, and endothols are used more for SAD control, but they can be used for green algae. So that's just understanding there are 11 different products here to look at. So let's just look at some efficacy data. So then we did them at 4 different concentrations. So, but all within label rate. So all within legal application rate. So next slide, please. So again, just to make it easier for you to see, what's in red is peroxides, and the other ones are coppers except for one, which is the hydrothol all the way to the left. So the first thing you see, hydrothol, which is good for green algae and filamentous greens, is didn't really work that well compared to other ones for cyanobacterial control. But when you go through there, you see that on the G's, way on the right, you see that there is a different effect with granular peroxide and liquid peroxide. But here, just remembering here, we did this within legal application rate. You can legally apply liquid peroxide at higher concentrations than you can granular peroxide. So if you go to the all to the right, you see that the maximum on the granular was 10 milligrams of peroxide per liter, but the maximum for the liquid was 22. So if you take that and into account, they are as effective, they are comparable to each other. But again, like for example, if you look at the coppers, they work extremely well, even at the lowest concentration of 2 milligrams per at 0.2. So again, the biggest thing is here is see that formulations have a little bit of difference of effects. Okay, next, and then if you look on the top, if the top right, you can see an example of bleaching, which is untreated control, different concentrations when you see at the higher rates. And if you look at this was the liquid peroxide, and you can see how it does bleach and how it does work. Can you next slide? So a big question afterwards is, okay, what about toxins, right? Well, of course, because the idea is that peroxides can degrade toxins. But if you look and without releasing them, or they release and degrade. But if you look at the in the red on the bottom right, that's percent of aqueous microcystins to total microcystin concentration. And the peroxides really lyse those cells because they affect organic material, but they allow those microcystins to be in the environment. Okay, they will decay over time. So I found that odd. It could be several things because this was done in a beaker, but I'll show you some other data as well. So we could have killed all the bacteria that degrade microcystin as well. And this is focused on microcystin. But if you look at some of the other non-red aspects, just if you look at, for example, the algae myosin, the top, let's say the highest one in that bottom right graph, you see there's very little release of toxin. So in this case, what you do see is a death or senescence or control of a bloom with very little change with very little lysis of the cell. So that leaky cell hypothesis that is always talked about with control of algae, it's not always there. But this is at low concentrations of this copper. Okay. And again, what you saw here on the top was microcystin concentrations over time. But again, even though some of the toxins released at the beginning, over 9 days, we saw almost total decrease of microcystin. So at the end, even at 4 days, yes, it did control differently, but we did have control of bloom and control of and decrease of toxins. Next slide. So taking that, I took out the slides that had liminal corrals. So there was a middle step. So we took these and we went in the field. So this is Pahokee Marina, looking at mid-rate granular peroxide control. Okay, so this is we looked at microbial community, which I told you guys before is important, and the habit itself. Looking at 5 milligrams per liter of peroxide, which is half the maximum label rate. So next slide. So this was done in several parts of that marina, and as you know, blooms can move around, but the the red on the pinkish red, I guess, I hope nobody is colorblind because those aren't really very friendly colorblind colors. You see the 0 hours after treatment. So on the left, you see the concentration of chlorophyll. I mean, the vertical axis, you see the concentration of chlorophyll or phycocyanin or cell counts. So those are all three measures of abundance. And on the horizontal line, you see hours after treatment, okay? And that goes from 0 to 48. So in all aspects after treating, you see within 4 hours a significant decrease of abundance. So it worked. There was a huge variation in the control because we're looking at points around the dock, the marina that some had bloom, some didn't have bloom, if that makes sense, right? We still, those were the points. So even though one had a bloom, the other one didn't, we still had to count that, right? We still had to take those samples in these fixed points. So next slide. And then we're going to, again, so that's the question. So it worked wonderful. The bloom was was controlled. And in this case, the question is microcystins. So just take a look. The total microcystins, you can see that's 90, and the aqueous microcystins is at 20. So at the beginning, you saw about over, it could be over 90 ppb of toxins. 8 is the recreational guideline by the EPA. So you see there. So very much, very out of this recreational guideline. But then you see after 4 hours after treatment, 24 and 48, you really see a humongous decrease to, let's say, almost 8, at 24, about 8, which is the recreational guideline at the EPA level. But you do see that when it was treated, it all became aqueous microcystins. Okay, mostly if you look at the other side. So yes, you did see a control, but you did see a change to aqueous, but you saw an overall decrease. And why, if you don't understand why, and I didn't say why am I talking about aqueous in total, is that the aqueous form is much more difficult to control when it gets in drinking water. Because if you guys, I don't know if you've worked with drinking water, but the drinking water systems, as you flocculate, you do all that you try to gather that biomass and remove it without lysing the cell, because when you lyse the cell, toxins are in there and you can't remove it that way. You have to remove it with activated charcoal, and that costs a lot of money to do. So, so in drinking water, it's very important to treat without transferring or making the compounds become aqueous. In recreational waters, let me go back. Not that it doesn't matter. It is that the recreational guidelines are on total. Okay, if that makes sense. Anyway, so next slide. So then I wanted to bring in some of these novel things that you may hear, and I wanted to focus on nanobubble because it is being used a lot in Florida. So is ultrasound, but the DEP has funded some work in this, and maybe you guys have as well. So we had an Army Corps grant with Ohio State University with a couple, with Heather Raymond, Linda Weavers, Rachel Gabor, and Justin Chafin, looking at controlling, looking at algaecides and also ozone nanobubble. So in this case, as you can see, not to get into, this is replicated mesocosms, and look at you can see the photos. You're looking at a comparison between copper algicide, peroxide algicide, low NBOT and high NBOT. So those are pictures from on top of the tank with a Secchi disc. And you do see, for example, that control is beautiful. Copper, in this case, did not work that well, or it's taking longer to work because the water was a little bit cold. The peroxide, you start see bleaching, as you can see, because you can start seeing, but you really see that NBOT low and NBOT high really cleared up that water, but you also see all that crud on the bottom as well. But that's the technical term. It's called crud. But anyways, you see these that there is something going on. So this is visually, you see something going on. That was the first mesocosm treatment. Then let's go to the next slide. We'll look at another one. So in this case, with a second treatment, because after a couple of years, we really found out the ozone to DOC ratio is extremely important to understand why ozone nanobubble works. Okay, so in this case, we did the control. We had oxygen only, because you've also heard of oxygen nanobubbles without the ozone. And look how beautiful that bloom is. You know, the cyanobacteria really like those oxygen bubbles. But when you put ozone in there, then you start seeing effect. So in here you see a low, an intermediate, and a high dose of ozone, and that water really got clean. It was amazingly. Just to go see some graphs and not just pictures. This is the next slide. So this is the same data, but presented this way. So what you're looking at is the pink line is the control, the green line is oxygen only, the blue line is the low NBOT, yellow is medium, and orange is high. Okay, so and then you're seeing time on you're seeing time and total concentration of whatever you're looking at within each one of these graphs. So the first one is looking at chlorophyll, the graph A over time. So you can really see at medium and high NBOT. Actually, everything had an effect, but you see the ones that had ozone, in this case, really decreased much higher than oxygen only, and the control maintained. Phycocyanin is the indicator for cyanobacteria. So you did see a decrease of phycocyanin over time as well. And then on the bottom are both total and aqueous microsystems. You did see that correct and ozone nanobubbles also lysed all the cells and made all the toxins into an aqueous form. But over time, if you look at C, those did decrease, right? But there was a very high peak at the beginning. So that meant that the cells lysed. And if you look at the control, which is did I say pink? It looks like pink purple. That one, it was high the whole time, at 40 ppb here, for example. But then at the aqueous form, almost nothing. And I think I have one more slide or two. So just concluding this, there are many proactive and reactive management. I just gave you guys a couple seconds of a couple of them, but there's no silver bullet. And that's why we don't have a fix for everything. So we really need to think about integrated pest management. You think about that in agriculture, but we really need to think about that as in HAB control. So control methods may work in some waters, not others. There's a humongous question of scale and economics because, especially these bacterial products, a lot of them work wonderfully in aquaria, but when you put them out in a HOA or Lake Okeechobee, for example, or, they're not working, right? Or they may work a little bit. So this, again, I'm not saying anything against any product, but really, that the scale is important. And of course, I heard Jennifer talking about economics and finances and grants. So really, if you're reviewing papers on proposal in this, I would suggest also to have that question of, is this feasible in the system they're proposing? Not that it's not feasible or not or that it can't work. Just maybe you need to look at a different system. Not all waters are the same. Not all cyanobacteria and algae are the same. Like, for example, what I talked to you about peroxidases. So the efficacy of treatment methods differ due to target species, water temperature, water chemistry, formulation of compounds, and many other aspects. So the last slide is coming up. Just a thank you to all. And remembering, these are the people that, I'm here talking to you, but these are the data of the people in our labs that do the work. And funding for the different aspects of this work are there to thank them. And if anyone, I took out this slide too, but if anyone wants to go to a week full of HAB Science in Cedar Rapids, Iowa, in October, please, we have our annual or biannual meeting, and Barb's in there. So Barb, please, you can talk to Barb. She's I'm co-chair of the National HAB meeting committee, and she was one of the founding co-chairs. So she can tell you all about it. And she's from the Midwest. So Barb, I'm giving you a little bit of there. So just go to her. Okay. Thank you so much, Jennifer, and everyone. And thank you for listening to me.
Thank you.
And I think I did it in 20 minutes, right? I forgot the timer. Yeah.
Thank you, Dail, for all that information and considerations to consider when mitigating for blue-green algae. It's really helpful and informative. So unfortunately, we don't have time for questions, but I really encourage you to email Dail. Can you maybe go back to his email real quick? If you guys want to write it down and ask him questions, I think that would be a great follow-up for you guys.
Or you can Google Dail Laughinghouse. I'm one of the only, well, there's 4 of us because I'm the 4th, but the only one working with HABs. two are dead, and one has humongous farms. So that's my father. So anyways, so I'm the one that if you want sod or tobacco, you talk to my father. If you want HAB, you talk to me.
Thank you.
Okay, thank you so much, and everyone have a nice day, and thank you so much for allowing me. And hopefully, I can meet some of you guys in person next time I'm on your coast. Okay, have a wonderful meeting. Thank you.
All right, so we're going to move to our next agenda item. It's a presentation of blue-green mitigation strategies and technologies. And this will be given by Dail Laughinghouse, who is with the University of Florida.